Hey everyone, welcome to Professor Long's lectures in microbiology. I'm Professor Bob Long. Now the videos that I'm shooting are intended for students to gain a very basic and fundamental understanding of some of the basic biological and processes in microbiology, so that as we go through this course or series of lectures, you guys get a very good understanding of what's happening in microbes and how they function. This is really important for people who are in the health science majors, the allied health science majors like nursing and OTA and PTA and so on, respiratory therapy, surgical technology and other things, so that you guys can understand what the basis for pathology is in many of the diseases that we're going to be treating in healthcare, and also how to prevent them with asepsis and aseptic technique and sterilizing things. So our goal is to understand some of the basic fundamental concepts in a really simple form, and then over time we'll make it more and more complex so that it all makes sense. Now, if you're watching this in YouTube land, please make sure that you learn the information that your instructor wants you to know, and any and all the definitions and terminology that they go over. While I'm not doing a complete review of all the concepts and all the material that you should know, I'm doing what I think is important for the students who are enrolled in my class, and for that all students should understand, and then you can plug in more detail and more analysis into that and understand an even bigger picture. Microbiology is a very complex topic, and we could talk for years about it and still not understand it. This is at the sophomore level, so this is an introduction to microbiology, so I'm going to keep it simple for now. If you like my videos and you find them helpful, please hit like, leave me a comment, and subscribe so that you get updates when new videos are loaded. And these videos are very rudimentary. It's me with a cell phone and a marker board. I'll try to make them much more fancy and slick in the future, but I'm doing this in a pinch. So, the last video we were talking about microbial metabolism, and we really didn't get into microbial metabolism. We set ourselves up for understanding it. We talked a lot about enzymes and what they are and what they do. They're proteins, usually, that increase the rate of chemical reactions or lower the energy of activation of many of the chemical reactions that occur in living organisms. We talked a little bit about coenzymes and cofactors and other things that affect enzymes, temperature, pH, and some other things. We're going to take advantage of all of that in the future to understand how we can regulate microbial growth. If we want to increase their growth or decrease their growth rate, what conditions must we provide them? Now, one of the things we're talking about in metabolism is we're talking about energy metabolism, the production of ATP. And I talked in the last video about what ATP is. Now, essentially, what we're going to do is we're going to go over a few definitions, but we're really going to draw out a lot of the chemistry of microbial metabolism. And in order to understand microbial metabolism, I want you guys to also understand eukaryotic cell metabolism, because there's a major difference between what prokaryotes do and what eukaryotes do. Now, when we talk about energy metabolism, we're talking about the production of ATP, adenosine triphosphate. Eukaryotic cells do that in 3 steps: glycolysis, which occurs in the cytosol, and then the Krebs cycle and the what we call chemiosmosis, or the electron transport chain, or oxidative phosphorylation. It goes by a bunch of different names, but it's the electron transport system, or the electron transport chain. And the last 2 happen inside the mitochondrion, which is a membranous organelle. Bacteria lack membranous organelles, so they don't have mitochondria, but they can still do some forms of aerobic respiration. Not all bacteria do. It depends on the bacterium and the membrane and the enzymes in that membrane. So which enzymes a bacterium or a cell has can determine how it produces energy. And we're going to talk about this in some detail. Now, as a real brief overview of what's going to happen is this. There are 4 organic compounds we're going to talk about. We talked about this in the last video, and I'm going to list them as 1, 2, 3, 4. 1 is carbs, carbohydrates. And we digest these with saccharidases to release glucose, C6H12O6. Another one would be proteins, And we can digest this into amino acids. And what I really want to do is use amino acids to synthesize proteins in my cell. But when I'm out of sugar, I can convert proteins into, or amino acids into glucose. A third one would be these compounds called triglycerides. Triglycerides and this is not necessarily the order that I taught them to you in before. I usually do lipids second and then proteins. And let's do that. Let me rearrange this a little bit. I'm sorry for that. But the second one we call lipids. And we can digest some lipids into what we call triglycerides. There's a certain class of lipids molecules called triglycerides. And then the triglycerides can be further digested into fatty acids plus glycerol. And these two compounds, the fatty acids and glycerol, can be converted into glucose if necessary. The third major class of organic compounds we talked about were proteins. And by the way, these would be called lipases here. Proteins can be digested by proteases and peptidases. I hope you can read this on your screen. If not, you'll have to magnify the image. But this is a review of what we did last time. Into amino acids. Now my goal is to use amino acids to synthesize new proteins, to renew all the proteins inside the cell. But if I'm out of sugar and I'm out of triglycerides, one of the things I can do is I can send amino acids, convert them into glucose. And I'm not even going to talk about the nucleic acids, because if we have to go there, we're going to die. We can't afford to do that. So, now, the importance of glucose is that glucose can be converted into what we call two molecules of pyruvate. Now, when we do this conversion of glucose into two molecules of pyruvate, essentially what's going to happen is I'm going to get some energy out of this. And this process is known as glycolysis. Glycolysis is the breakdown of glucose into two molecules of pyruvate. And one of the things that's going to happen in this process is as I break this down, I'm going to get some energy out of it. I'm going to get two molecules of ATP, and I'm going to get these other molecules called NADH. I'm going to get two of those. Let me rewrite that. I'm going to get two molecules of NADH. So in the process of glycolysis, one of the three major steps in energy metabolism in our cells, I'm going to convert sugar into two molecules of pyruvate. I'm going to get two ATP into NADH. And this is going to occur inside the cytosol of our cells because that's where the enzymes are. Now, what's going to happen with the pyruvate is if I'm going to look at a eukaryotic cell, in a eukaryote, I'm going to have an organelle that has two membranes, an outer and an inner membrane. And the inner membrane, the inner mitochondrial membrane, so this organelle is called the mitochondrion in a eukaryote. We have the matrix inside and then the inner membrane. The inner membrane of a mitochondrion has all these enzymes linked together in little groups of 6, all inside of here. And they're called cytochromes. And they're on the folds of the inner mitochondrial membrane called cristae. So here's what happens. In eukaryotic cells, this waste molecule called pyruvate can be run into the mitochondrion. It's going to be converted to a stuff called acetyl-CoA, or acetyl coenzyme A, and then it goes around in this circular group of lots of chemical reactions. And this process right here, I'm going to put a little asterisk, is called the Krebs Cycle, after Sir Hans Krebs, who first described it and figured out the chemistry. It's also called the TCA cycle. TCA stands for carboxylic acid. There's the T, C, and A. Because this acid has 3 carboxyl groups on it. Last time we talked about what a carboxyl group is, the C double bond O with an OH. And it happens to also be called citric acid. So it's also called the citric acid cycle. And this process is going to take the pyruvates that are left over, convert it into acetyl-CoA, and run it through a series of chemical reactions. In those chemical reactions, I'm going to get some things out of it. And these are the things that we need to know. So this is the second major step in our energy production in a eukaryotic cell. The Krebs cycle, or the TCA. I have glycolysis, I have TCA, or Krebs cycle. And what I'm going to get out of the mitochondrion here is I'm going to get 2 molecules of ATP. Each pyruvate has to go in and go around and give me 1 molecule of ATP, but because I had 2 pyruvates, then I get 2 ATP. I'm going to get some carbon dioxide. So I can produce carbon dioxide here. This is where the carbon dioxide that we exhale comes from. And I'm going to get some other molecules called, and they're not really going to come out here, I'm going to get other molecules called NADH, and I'm going to get 6 of those, not that I care about the numbers. And you're going to get these other molecules called FADH+. I need to make my mitochondria a little bit bigger. So this mitochondria is going to be funky in shape, but nonetheless you get the idea. So as I go around my TCA cycle here, I'm going to get 2 molecules, or 6 molecules of NADH, and 2 molecules of a stuff called FADH+, or FADH2, I should say. Okay. So, if I look at my energy metabolism, there's my cytochromes. If I look at my energy metabolism, I've essentially taken the leftovers of glycolysis and doubled my ATP production. I get a total of 2 net ATP. Now, technically, I produce 4 ATP here, but it takes 2 ATP to get it going. So since I burn 2 and I produce 4, and I do the math, I get 2 net ATP gained. And I get these guys called electron carriers. I take the waste of glycolysis and I can convert that in the presence of oxygen. If I'm talking about aerobic metabolism, aerobic metabolism means oxygen is present. In aerobic metabolism, I'm going to convert the pyruvate inside the mitochondrial matrix into acetyl-CoA and go around this chemical reaction called the Krebs cycle. It's a series of steps we don't need to get into, also called the TCA, or citric acid cycle. And I'm going to produce 6 NADH and 2 FADH2. Again, these are called electron carriers because they're going to carry electrons to the electron transport chain, or the cytochromes in here. I'm going to get 2 more ATP, and I'm going to get my carbon dioxide. And I'm talking about energy metabolism, so I'm counting my ATP. 2 here, and now I've doubled my energy production. And here's the kicker in eukaryotic cells. Okay? All of these electron carriers, the NADH from glycolysis, the NADH from Krebs, and the FADH2 are going to carry electrons to these little cytochromes, or enzymes, and these enzymes are going to rip the electrons off and pass them from one to the other in a series of reactions. And electrons are simply a lot of energy. And what it's going to do is it's going to take a molecule of oxygen and some protons, and it's going to convert those into water. Remember, a proton is simply, or a hydrogen atom is a proton and 1 electron. And if I can take these electrons, combine them with these protons in a proton pump, it's actually called ATP synthase. It's going to use oxygen, protons, and electrons to form water. And in that process, that little ATP synthase is going to kick out another 32 ATP. And when I do my math, I'm going to get a total of 36 ATP from one molecule of glucose. 2 in glycolysis, 2 more in Krebs cycle, 1 per pyruvate. And then this little process right here is called the electron transport chain. So some people call it the ETC, others call it the ETS, and we can change the word chain for system. I can't write at the bottom of the board. This is our third step in energy production in our cells. The electron transport chain, because I'm going to use oxygen to add a phosphate to ADPs forming ATP, is also called oxidative phosphorylation. Oxidative phosphorylation is the electron transport chain. It's also called chemiosmosis because there's some osmosis going on here, but we're not going to get into that. Okay? And this last guy in the chain here, all of these get different names, but the last enzyme in the process called ATP synthase is also known as a proton pump. And it's going to pump protons from one area of the cell to the other, combine those protons with electrons and oxygen to form water, and when it does that, it takes an ADP and sticks it with a phosphate and forms ATP. And it does that 32 times. There's so many per each electron from each one of these, but I don't care about that mathematics. So now, ultimately, there's 2 things going on here. My 4 major organic compounds, and I've only listed 3. We didn't put nucleic acids because I don't really care about them at this point, at least for this topic. But carbohydrates are broken down by saccharidases to give us glucose. Lipases break down lipids, and when we get triglycerides, we can break those down into fatty acids and glycerol and convert those to energy. And if I need to, I can convert amino acids into glucose. Now, one of the kickers for the amino acids is that in order to do that, I actually have to kick off a group of amino groups, and when I remove the amino group, we call that deamination, or to deaminate, remove the amino group. And that's going to give me some H2N, or NH2, which is a negative charge. If I combine that with some protons, eventually I get NH3, which is ammonia. And there's some tests that we can do because some bacteria can convert amino acids into sugar. And when they do that, we can test for the ammonia or the nitrogen-containing compounds that they produce. And we'll talk about those tests when we do this stuff in lab. So this drawing is rather a little bit confusing considering the small space of board I have to work in. So the first part is the organic compounds can all be converted into glucose. When I do it with amino acids, I have to deaminate and remove the amino group. Now when I erase all that part, okay, I'm just going to erase this. Now we know the source of glucose can come from carbohydrates, lipids, or amino acids from proteins. From proteins I have to deaminate. But once I get down to glucose, in the cytosol of the cell, I can do glycolysis, the breakdown of glucose into 2 molecules of pyruvate. And that's going to give me 2 ATP and some electron carriers, or NADH. Then I can take the leftovers of that, and under aerobic metabolism, I can convert the pyruvate into acetyl-CoA inside the mitochondrion, and then I can run that through a series of chemical reactions called the Krebs cycle, the TCA cycle, and the citric acid cycle. I'm going to get 2 more ATP, I'll get some carbon dioxide, and I'll get 6 electron carriers in the form of NADH, and 2 more in the form of FADH2. That will take all of these leftovers, all of these electron carriers, and let me run those to the cytochromes, the enzymes on the inner mitochondrial membrane, and then I'm going to perform the electron transport system, or the electron transport chain, also called oxidative phosphorylation, because it's going to use oxygen to phosphorylate ADP into ATP. In order to do that, I combine the electrons and some protons to form hydrogen, and I take the hydrogens and the oxygens and convert them into water, and I get 32 ATP. And because oxygen is the final electron carrier, we call this aerobic metabolism. So, let me erase this and rewrite it a little bit neater, but what we call aerobic metabolism uses oxygen as the final electron acceptor. And aerobic metabolism can happen in some prokaryotes. They just don't have the mitochondria. The eukaryotic cell has the mitochondrion, and that's where all of this is going to occur. So some things I want you to walk away from knowing. Number one, glucose is converted into 2-pyruvate in a series of chemical reactions called glycolysis. We don't need to go over the individual steps right now. This occurs in the cytosol of the cell because that's where the enzymes are that do it. It gives us 2 ATP and 2 electron carriers in the form of NADH. The pyruvate moves into the mitochondria. It gets transported into the mitochondria, and inside the mitochondrial matrix, we're going to convert it into acetyl coenzyme A and do what's called the Krebs cycle, the TCA cycle, or the citric acid cycle. It's a series of chemical reactions that goes from acetyl-CoA back to acetyl-CoA. In the process, I'm going to get some carbon dioxide as waste. I'm going to get 2 more ATP, doubling my energy production to 4 total. And I'm going to get a bunch more electron carriers, 6 NADH and 2 FADH2. The NAD stands for nicotinamide adenine dinucleotide, and the FADH comes from fumaric acid or fumarate, and it's an adenine dinucleotide, and we don't really care about that right now. But what I do care about is that these leftovers, all of these electron carriers, go to the cytochromes on the inner mitochondrial membrane, the folds there called the cristae, and these are the enzymes, the cytochromes are the enzymes, that rip the electrons off of here and pass them from one to the other, and then the last one will combine them with hydrogen and oxygen, and these protons get converted with oxygen, get converted into water. And that ATP synthase at the end is a proton pump that is going to convert ADP into ATP, and it does that 32 times for each oxygen molecule, or for the number of electron carriers that I have, not for each oxygen molecule. Okay? So that's a lot of information, and I've repeated it again and again and again so that you guys learn to say this and teach this. You should be able to draw this little map. If you can't, you practice it. If you can do nothing else, draw this part over and over and over again. Then draw this part over and over and over again. Then add the last step. Know the 3 steps of energy metabolism: glycolysis, Krebs, TCA, citric acid cycle, and the electron transport system, or oxidative phosphorylation. Okay? Now that's called aerobic metabolism because it requires oxygen as the final electron acceptor. There are some organisms that don't use oxygen. They can use nitrogen or sulfur, and that would be called aerobic metabolism. Well, it's called anaerobic metabolism, but we can still get a lot more ATP out. We just don't use oxygen. We'll talk about that in the future. Now, what happens if there is no oxygen present, even in our eukaryotic cells? If these cells don't have oxygen here, then these electrons can't be transported, and everything starts to shut down and back up. And what happens in that situation is this. If there's no oxygen present, then the pyruvate gets converted to a stuff called acetaldehyde. Well, we're just going to skip a step. It gets converted into what we call lactic acid. So when I'm breaking down glucose to make energy, ATP in my cells, if I do it with not enough oxygen present, I can't convert the pyruvate into acetyl-CoA and finish this process. I simply kick it over and make lactic acid. And lactic acid is what causes our muscles to be sore. So when you're working out, if you're working out or running harder and faster than your heart can pump the oxygen to your cells, then your body will start to slowly build up lactic acid, and your muscles will be sore later on in the next day. So we call this process of converting pyruvate into lactic acid without oxygen, it's called fermentation. Our cells can do fermentation. They can convert pyruvate into lactic acid. Now, all of this is important because now we can see, how do bacteria differ? Some bacteria can do fermentation, but they're not going to produce lactic acid usually. Bacteria convert pyruvate into ethanol, alcohol, or some yeast can do that, I should say. And some bacteria can convert this into acetic acid as well. And we'll talk about these processes, how microbes can convert pyruvate into ethanol or acetic acid through the process of fermentation. And some microbes can actually do this process of sort of the electron transport, or what we call oxidative phosphorylation, but they don't use oxygen as the final electron carrier. So it's not really an aerobic metabolism. And some microbes do that, only they use nitrogen or sulfur as the final electron acceptor instead of oxygen. So it would not necessarily be aerobic metabolism because we're not using oxygen as the final electron acceptor. Now this is a big deal. So I hope you have this down in your brain. Now, I'm going to redo some of this again in a little bit. Let me erase all of this. We'll do it again, and the more that we do it, the more it should stick to your brain. But then we're going to put in the differences that we need to see between certain organisms. Now, before I go into all of that, that's going to set us up for understanding some of the terminology that I want you guys to know that are in my notes. Number one, you should know the 3 important steps of ATP production in eukaryotes, and some prokaryotes can do these steps. Okay. Aerobic metabolism simply means that it uses oxygen as the final electron acceptor. When I rip these electrons off and rearrange these molecules, I'm releasing energy to form ATP, and the final electron acceptor, if it's oxygen, then the process is called aerobic metabolism. If the process is called anaerobic metabolism, then it uses some molecule other than oxygen as its final electron acceptor. And I'm simply using some notes because I just typed these up for my students, and I want to make sure I'm going in the order that my students need to know this. A lot of times it's nitrate or some nitrate-containing compound. Sometimes it's a sulfur-containing compound or sulfates. What organelle do we know plays a major role in the large amount of ATP produced in eukaryotic cells? It's mitochondria. Do bacteria have mitochondria? The answer is no, they don't. They're prokaryotes. They don't have membrane-bound organelles. Archaea and bacteria, those 2 domains, do not have mitochondria. They're prokaryotes. All the eukaryotes, fungi, protozoa, animalia, and plants, animals, fungi, funguses, and protozoans all have mitochondria because they're eukaryotic cells and they can all undergo this process. Okay. Now, some things that you need to know about glycolysis. Where does it occur? It occurs in the cytosol of the cell. Why? Because that's where the enzymes that perform glycolysis are found. And we don't need to know all of those enzymes. We don't want to go through glucose and glucose 6-phosphate, then fructose 1,6-bisphosphate and all those steps. We don't need to do all that stuff. That's for a biochemistry class. Does glycolysis require energy to be activated? Yes, it does. Now here's a little detail that I did not put in previously. If I'm going to undergo glycolysis, so here's my molecule of glucose. And when I undergo glycolysis, in the cytosol, then all of the enzymes that are going to perform these chemical reactions, it's actually going to take 2 molecules of ATP to make it go. It actually produces 4 molecules of ATP. And if you do the math, 4 minus 2 is going to give us 2 ATP as a net gain. We grossed 4. But when I take away the 2 it took to get it to go, I get a net gain of 2 ATP. If you don't understand net and gross, next time you get a paycheck, look at your gross pay, and that's how much you made. Let's say it's $5,000, and then what they put in your bank account, let's say $3,000 or $3,500, that's your net pay. The rest went to the government and taxes and everything else, which if you ever get to see your paycheck, it's alarming how much they take. So it takes 2 to get started. We produce 4. 4 minus 2 gives us a net gain of 2 ATP. The other thing that what I told you it gives us is these molecules called NADH. Now, where those come from is a molecule called NAD+. NAD+ is simply missing an electron. It's got a proton hanging on with no electron. When I rip some electrons off of these molecules in the rearranging of all these carbon atoms in here to give me 2 molecules of pyruvate, pyruvate can also be referred to as pyruvic acid. Okay? This is a little side note, but I want to tell you this. When I talk about an acid, it usually has some carboxyl group on it. Like if I were to draw carboxylic acid, if I take this carbon, and by the way, this could be some other part of a molecule, and I have my double bond O and this, plus the hydrogen ion that it's released, the proton, making it an acid, then this is called carboxylate. Anytime you have the term ate, it means the negative ion of the acid. Or it's the acid without the proton that it had attached. It's released the proton. So that's why these are called acids, is because they release their hydrogen ion, the definition of an acid, and become an ate. So if I say pyruvate, it's really pyruvic acid that gave off its hydrogen atoms. Now, another thing that's kind of interesting to note, I'm not going to test you on that part, but you should know that, is that the pyruvic acid, or pyruvate, is only made up of 3 carbons. So essentially I've taken a 6-carbon molecule, glucose, and rearranged it and then broke it down into 2 3-carbon molecules. That whole process is called glycolysis. It's going to net me 2 ATP. Okay? Now, so when I ask the question, does glycolysis require energy to occur? Yes, it does, and it requires 2 molecules of ATP. Now, during glycolysis, we take a molecule of glucose, we convert it into 2 molecules of pyruvic acid, and we get 2 molecules of ATP as a net gain. Now, for the rest of metabolism, it gets a little bit crazy from here. So, I'm going to redo some of this again, but I'm going to show you the difference between eukaryotic cells and prokaryotic cells. And part of the reason that I'm doing all of this over and over and over again is because the more you see it, hopefully the more it sticks to your brain. So now, I'm going to do the whole process. I'm going to take a molecule of glucose. That's the wrong marker. I don't like the way this one writes. I need to get rid of it. Take my molecule of glucose. It's going to enter my cell. This is going to be a eukaryote over here. As it enters the cell, I'm going to convert the glucose into 2 molecules of pyruvate. Well, I'll take the glucose, and as it enters the cell, I'm going to convert it into 2 molecules of pyruvate. Now, what I want you to note is there's a couple of things that happen that are important. As I run it through the chemical reactions to turn it into pyruvate, in this process, it's called glycolysis. I'm going to get 2 molecules of ATP as a net gain, and I'm going to get some electron carriers called NADH. I'm going to get 2 of those. Now, what happens to the pyruvate is the pyruvate is going to enter the mitochondrion. As it enters the matrix of the mitochondrion, it's converted into another molecule called acetyl-CoA, or acetyl coenzyme A. It runs through a series of chemical reactions, a whole bunch of steps: citrate, isocitrate, malate, fumarate, alpha-ketoglutarate, oxaloacetate, I forget the order. But nonetheless, this is called the Krebs cycle. Also called TCA, or citric acid cycle. And the whole point of that is it's going to give us 2 more molecules of ATP. It's going to give us some more electron carriers. I'm going to spit these out over here. I'm going to get 6 NADH and 2 FADH2. I get 3 NADH per pyruvate, 1 FADH2 per pyruvate. And one of the things it's going to spit out in the process is that's where we're going to get carbon dioxide. And then finally, the last step. So here's the 3 steps. 1, glycolysis. 2, Krebs cycle. And in the 3rd step, I'm going to send all these electron carriers to my series of cytochromes. And these cytochromes are the enzymes that are in the inner mitochondrial membrane, and they're on the folds called cristae. And essentially, I take the electron carriers, NADH and FADH2, these NADH, and these FADH2 are going to give electrons from one to the next in a series of steps. And in the process, I'm going to take oxygen from outside the cell that I inhaled. I'm going to take some protons that are in here, and I'm going to pump them through, and I'm going to form water using the electrons, some protons, and oxygen. And this last enzyme, called ATP synthase, is called ATP synthase because it's going to take molecules of ADP, adenosine diphosphate, take a free phosphate molecule and attach it and convert that into 32 more ATP, giving me a total of 36. 2 plus 2 is 4, plus 32 would be 36. And this is the last step right here. It's called the electron transport chain, or ETS, or oxidative phosphorylation. It can go by all 3 names. Some people even call it chemiosmosis. That's the 3rd step. So in these 3 major steps, glycolysis, Krebs, TCA or citric acid cycle, and oxidative phosphorylation, or ETC or ETS, that's simply going to give me all of my ATP. Okay? Now, a little kicker here. I'm going to put, if oxygen is present, if I have enough oxygen going, then I can do all of this. But without the oxygen, this backs up, and I stop energy production, and I only get 2 ATP per molecule of glucose. Then what happens in this situation is that the pyruvate gets converted into lactic acid, or lactate. And that's the burn in your muscles. The enzyme that does this is called lactate dehydrogenase. And essentially what happens is it's going to rip some of these electrons off here and convert this back into NAD+. And then we can run back through the cycle, and we're not going to talk about that right now. Now that's in eukaryotes. This is the 2nd time we've done it. Try to do it a little neater. But what I wanted to do was leave a little room over here, because my next cell that I'm going to draw is going to be a prokaryote. My prokaryote is going to be much simpler. My prokaryote does not have mitochondria. So what happens in prokaryotes is that they're going to take glucose in, and when they get the glucose in, they're going to convert glucose into 2 molecules of pyruvate. Out of that, they're going to get 2 molecules of ATP as a net gain. They get some energy. They're also going to spit out NADH. Now remember, if I were to draw a hydrogen atom, it's 1 proton in the nucleus and 1 electron. So when I ionize hydrogen, if I rip off the electron, I get a proton. If I add an electron back in, I get a hydrogen atom. So what happens is there's a molecule called NAD+, nicotinamide adenine dinucleotide, that's going to come by here and it's going to collect any extra electrons. Have you ever ripped something apart and put it back together once it's fixed, and there's like a spring and 2 screws left and maybe 1 washer, and you're like, I don't know where that goes? I'll just put them in a drawer, and hopefully later, hopefully my gas grill doesn't explode, or whatever it is that you fixed. Well, when we rip apart glucose to convert it into pyruvic acid or pyruvate, I have some extra electrons, and those electrons get combined with nicotinamide adenine dinucleotide, and when I stick the electron onto this proton, it turns into NADH. It forms a hydrogen atom. Now, because prokaryotic cells are lacking mitochondria, what do they do with the pyruvate? Well, the pyruvate is going to get converted to a stuff called acetaldehyde, and then that's going to get converted into ethanol, which we can also call ethyl alcohol. Oops, misspelled alcohol. Ethanol, or ethyl alcohol, is the byproduct of this process. And essentially what happens in this process is I take the NADH and I convert it into NAD plus and take the electrons off, and we convert acetaldehyde into ethanol. Now I'm not really concerned about all of that, but it is a way to show you that when I do this process of converting pyruvate into ethanol, it's going to generate the NAD plus. This process, by the way, is called fermentation. That's the same thing that happens in our eukaryotic cells when we don't have oxygen present, as we get fermentation. Well, we convert the pyruvate into lactic acid using a different enzyme. So the enzymes that are involved in this process are present in bacteria, and they will, and yeast, which is a type of fungus, that will convert it into ethanol. So fermentation in prokaryotic cells gives us a different end result. Instead of lactic acid, they get ethyl alcohol. And this is what causes us to make things like beer, wine, cheeses, certain breads, yogurt, things like sauerkraut, misspelled sauerkraut, but pickles. Because in some instances, they can even convert this to acetic acid, and that acetate or acetic acid is what can pickle things, or we use the vinegar directly sometimes. But nonetheless, in a prokaryotic cell, I'm going to get glycolysis. And it's going to produce 2 ATP. Notice there's no energy production in fermentation. The point of fermentation is not energy production. The point is it's going to actually form NAD+. It's going to convert the NADH to NAD+, which allows us to do more glycolysis as long as sugar is present. So no energy production in fermentation, it just recycles the NADH so that I can do another round of glycolysis and I can keep converting these things. The fermentation produces NAD+, which we use for glycolysis to make NADH. We use the NADH in fermentation to make NAD+, and we get alcohol out of this process. And this is how we use microorganisms to make beer and wine and certain cheeses and certain breads and things like yogurt and sauerkraut and pickles. It can also cause spoilage of milk and the souring of milk. We make cottage cheese. So we can use bacteria and microbes for some industrial purposes and for food production. Now, understanding all of this stuff is really important because later on we're going to take advantage of all of these things. Now, I guess one of the things that I want to talk about, or at least I want to mention here, is one of the bacteria that are, not bacteria, a yeast that is involved in this process of fermentation is called Saccharomyces cerevisiae, or cerevisiae. Now, Saccharomyces cerevisiae uses sugar, that's the saccharo part of the name. Cerveza, if you speak Spanish, is beer, and it's used in beer production. It also produces wine, it also produces the ethanol that is involved in a number of these other things, okay? And in some of these organisms, we get some carbon dioxide out of this process as well. And that carbon dioxide is used as the gas that produces the leavening of breads and causes bread to rise, so to speak. Now, one of the things that's important is that in prokaryotes, we don't get the Krebs cycle, and we don't get the oxidative phosphorylation, or ETS, out of the mitochondria. Although we can do it in some bacteria. Now, a couple of things I do want to briefly mention. I want to refer to my notes so I don't forget this. There's a couple of bacteria that can do this, that can provide lactic acid. And there's a test that we can use to see if there are some bacteria present causing some problems. And so one of the bacteria that can perform the formation of lactic acid is one that's good for our gut called lactobacillus. And lactobacillus is one of the things that we sometimes call a probiotic. If you look at certain probiotic yogurts, the ones that have cultures, live cultures in them, they use lactobacillus. And those lactobacillus are actually really, really good for your gut and certain breakdown of certain compounds in our gut, and they're really important in digestive health. Now, another bacterium that can perform this chemical reaction due to lactate dehydrogenase is a streptococcus. Streptococcus sometimes can create disease process. And we talked a little bit about streptococcus in our labs, and we'll talk about them in the future. So some of these streptococci can result in the formation of lactic acid, and they're involved in some health-related issues. We're not going to get into that until we actually get to that process, because that's a whole other lecture in ball of wax. I think that's everything that I want y'all to know about this process for this moment, okay? Now, there are a couple of bacteria that can do oxidative phosphorylation, but they do not use oxygen as their final electron acceptor. Now, let me look at my screen so I can make sure that my board is not... I'm going to move the image so I get a little bit more room on the left side over here. And I'm going to raise it up just a hair so that you guys can still see the top of the board. Okay, I'm sorry for this movement of the screen during the video, but it's just me. I'm a one-man operation. Okay. Now, one of the things that I want to show you is that we can actually get some of these extra steps that occur in certain microbes. And in this process, rather than using oxygen as the final acceptor, one of the differences between some of the microbes and eukaryotic cells, prokaryotes and eukaryotes, is that prokaryotes don't have mitochondria. But the prokaryotic cell membrane can have these enzymes out here. And they're not always the cytochromes. Some are called ubiquinones and a couple of other names. And if you look at it, it almost looks as if the inner mitochondrial membrane is almost identical to some of the bacterial membranes and prokaryotic membranes. Also, we know prokaryotes have a single circular chromosome and 70 S ribosomes instead of 80 S ribosomes. Svedberg units is the S, so they use different ribosomes for protein synthesis. Well, lo and behold, not only does mitochondria have a similar membrane, plasma membrane, with similar enzymes, they also have a single circular chromosome. If you've heard of mitochondrial DNA, it's a single circular chromosome like a prokaryote. And they have enzymes inside them that can do the Krebs cycle. There are a few bacteria and prokaryotes that can perform similar chemical reactions. And finally, the ribosomes inside mitochondria are 70 S ribosomes like bacteria. And to some scientists, to many of us, it appears as though eukaryotic cells ingested a prokaryotic cell and wrapped it with its own membrane, only it didn't lyse it with lysosomes, because this prokaryotic cell that entered a eukaryotic cell provided it with a lot of energy, and this cell got what it needed, so they lived in a symbiotic relationship. And it's thought to be part of the evolution of eukaryotic cells is that at some point they took an ancient prokaryotic cell that was able to perform some extra chemical reactions and convert the remains of glycolysis into more ATP, and they lived in a symbiotic relationship ever since. And it's considered part of our cellular evolution. Now, the only difference is, in some of the prokaryotes, the final electron acceptor is not oxygen. It can be nitrogen, or a nitrogen-containing compound, or a sulfur-containing compound. Now, we're going to get into some of those details in the next lecture. So, before I get off on any more of this information or have a really long video, I'm going to wrap this one up. So I hope you guys get to see that, number one, energy production in eukaryotic cells is a series of steps that require mitochondria, and if oxygen is present, we can take the pyruvate, convert it to acetyl-CoA through the Krebs cycle, get more energy, extra ATP, produce some carbon dioxide, and produce more of these electron carriers, and then those go into even more ATP production in what's called oxidative phosphorylation. Oxidative phosphorylation, producing water from oxygen and the electrons and some protons. If we don't have oxygen present, then the pyruvate gets converted into lactic acid in a process of fermentation. Not only can eukaryotic cells do this, but there's a bacterium that does this called lactobacillus, and those the lactobacillus, the prokaryotic cell that does this, is actually probiotic, and there's a few other organisms that can do this that we can test for that have some medical significance, clinical significance. We'll talk about that at a different time. Now, prokaryotic cells, since they lack the mitochondria, they simply convert the glucose to pyruvate, and then through fermentation, they convert it to acetaldehyde and ethanol, and yeast Saccharomyces cerevisiae does that and produces a lot of ethanol for beers and wines and cheeses, and also some prokaryotes are used to turn sugar into ethanol fuel, biofuels, and that was a popular thing for a while, but then we learned that biofuels gunk up engines and take massive fields of corn, and there's massive deforestation, and they may have done more harm than good. So we're moving away from some of the biofuels, and now we're moving more towards electric energy. Anyway, also, some of the prokaryotes have the enzymes out here that can perform similar steps to oxidative phosphorylation, only they don't use oxygen as the final electron acceptor. They can use nitrogen-containing compounds or sulfur-containing compounds. And again, we're going to talk about those in the next video. So, I hope that this made some sense to you. I hope that you see that microbial metabolism is different than most eukaryotic cell metabolism, but there are some eukaryotes that can do this still, that are single-celled organisms, protozoa, and then some yeasts, which are single-celled fungi, can do this process, okay? And in the absence of oxygen, they can undergo fermentation and produce lactic acid. Other prokaryotic cells undergo fermentation and produce ethanol, ethyl alcohol. There are a few that produce acetic acid, acetate, or vinegar, and there's even a few that produce a couple of other compounds like 2,3-butanediol and some mixed acids, and we'll talk about those and their clinical and medical significance at another time. So, I hope this was informative. I hope this provided you a basic foundation of some microbial metabolism without getting too much into the chemistry, and I hope that you learned something and had as much fun as I did. The next video will go in a little bit more depth and we'll wrap up microbial metabolism. Thanks for watching. See you on the flip side.